Okay, so I want to welcome everybody at today's DOE virtual seminar series. The star isotope today will be Cerium-134. I'm Veronica Mako from Los Alamos National Lab. I'm a scientist on the isotope production team. Today I will kick it off with a presentation on the production of Cerium-134, followed up by a couple more presentations on cerium. And I would like to point it out that if you have any questions, we'll answer that at the end. You can start typing them into the chat. All the question needs to go through the chat, and there will be a moderated Q&A session at the very end of after all these talks. So let's jump in and let's see if I can get to the next slide. So I want to briefly mention what was our motivation for Cerium-134. There is increased interest in application of targeted alpha therapy, and more specifically, there is big interest on actinium-225 and thorium-227. These are actinides, and as you can see, there is only a little bit from, I hope you can see my mouse on the screen, in part of periodic table of elements. These actinides sits right below lanthanides, and unfortunately, to use them, it's good to know the biological fate in the body. However, they don't have very good imaging handles. So our motivation was to suggest a new isotope, which is similar, Cerium-134. Its F element has similar chemistry, meaning the oxidation state and the coordination, and is somewhat longer lived, similar on the same timeline, several days, like the thorium-227 and actinium-225, to use as imaging companion for these emerging isotopes. As you can see, Cerium-134 decays in about 3 days to lanthanum-134, which the lanthanum actually emits the positron, so the system is called an in vivo generator. And on the cartoon on the left, you can see that indeed the actinium-3 and cerium-3 is somewhat similar size and oxidation state, but the cerium can be oxidized to cerium-4 and mimic the thorium, which is in 4+ oxidation state. So how to get to Cerium-134? How can we produce, and can we produce it with good specific activity, and can we produce it at high amounts useful for the usage for the end users? So in the beginning of the study, we looked at with our collaborators at the Berkeley National Lab, Lawrence Berkeley National Laboratory, at their 88-inch cyclotron at energies, how to produce the Cerium-134. And I should mention that this is proton-induced reaction on the natural lanthanum. And then here at Los Alamos, we have large accelerator, which goes all the way up to 100 mega electron volts proton. So we can look at the higher end of energy range to see what the cross-section for production of Cerium-134 look like. And you can see it on the plots displayed. And you can see that the threshold energy is about 50 mega electron volt. From that, the production increases, but then it falls off above about 70 mega electron volts. To mention that the optimum energy range is driven also by not only how much and maximize the Cerium-134, but also minimize other co-produced impurities. I cannot go into a lot of details, but there's 2 papers cited below. And I want to mention that at the higher energy range, some of the impurities, the cross sections are climbing up. So you can see in this kind of blue shaded area, this is the energy range we are using to produce cerium-134 here at Llano. So this was production. So how do, and then more theory, this is kind of practical, images to show you what it's look like. So we start with lanthanum metal, which is shaped into these nice lanthanum pucks. And currently we are using 32 grams of lanthanum metal, which is being irradiated and it's encapsulated. You can see this is the open encapsulation to visualize the lanthanum metal. This is what it's look like when it's all sealed, because the targets are sitting in cooling water, and lanthanum is fairly reactive metal and can oxidize easily, so it has to be encapsulated for the irradiation. There again is the incident and exit energy between 78 to 68 mega electron volts, and then the beam current of 100 microamps, and we irradiate for about a day. After the lanthanum is irradiated, it's brought up to our processing facility, where we open up the target, remove the lanthanum metal, and then lanthanum metal is dissolved in nitric acid, in 8 molar nitric acid. We bring the lanthanum into solution, and then we follow on with lanthanum cerium separation. Overall, lanthanide separations are fairly difficult, mainly for neighboring lanthanides. For cerium, it's, I think, the only one exception where cerium can be oxidized to cerium 4+, as I mentioned earlier, but also, and we do it with sodium bromate for the separation, but also the cerium in nitric acid media forms this anion, which is crucial for successful separation. So cerium forms anion and is retained on anion exchange resin, which is filled in a column. The lanthanum stays 3+ as a cation, passes through the column and gets washed off. Here on this plot, you can see the fraction volume versus yield, where we can track the lanthanum because lanthanum 140 gets co-produced. And then we wash off all the lanthanum, we wash off all the stable lanthanum, which is a huge amount. And then we can selectively elute the cerium-134. We remove the oxidation agent, and then we can elute the cerium with 50 millimolar nitric acid. This is really beautiful separation. It only takes one column, it's fast, and we obtain cerium in high yield. So what does cerium product look like? As I mentioned early on, when we irradiate lanthanum, there's actually a couple possible reactions going on. So you can see that other cerium isotopes can be co-produced, namely cerium-135 with less than a day half-life, cerium-137 metastable about day and a half, and then very small amount of cerium-139, which we try to minimize during production. You can see on the plot on the left, basically you start at the end of bombardment. At the end, we turn off the beam for several days, and you can see on the y-axis the activity of the isotopes produced. You can see that at the end of bombardment, there is actually a bit more of cerium-135, but that one decays really quickly. You can see what we do on the timeline. The isolation is happening between about the day 2 and 3. Then we perform qualification of the final product, and you can see at the time we qualify the product 4 days from the end of bombardment. You can see on the gamma spectrum on the right, on the top right, that there is still quite a bit of cerium-135. However, it drops down really quickly. So by the time we ship the product, there's almost no cerium-135. There is only tiny amount of cerium-137 metastable and small amount of 139. And you can see what the spectrum look like. It's a lot cleaner without the 135. Several days. This one was about 17 days, but it's very similar for about 10 to 12 days when we ship material to the customers. So what exactly the product look like? You can see in the right bottom corner the specification on the NIDC website. It has pretty high radionuclidic purity. As I mentioned, there is a small amount of the other cerium isotopes. There is limits what we have on these co-produced isotopes. Actually, at the time customer receives it, there is quite a bit less. And the specific activity is still very high, and we right now provide the cerium as 3+ in 0.1 molar hydrochloric acid, and the typical concentration is about 10 to 20 millicuries. However, the minimum is about 5. And I want to specifically mention that by inductively coupled plasma optical emission spectroscopy, we measure the total amount of cerium and lanthanum in the final product, which is important for labeling application. And you can see the total amount and the total concentration of the cerium and lanthanum in our products. These are data from our evaluation and first production batch here at LANL we produced. So of course, I have to mention like early studies on Cerium-134. This was in collaboration with Rebecca Abergel from Lawrence Berkeley National Laboratory, UC Berkeley. And with the early manuscript, which came out already 2 years ago, they applied Cerium-134 successfully in vivo and showed that indeed the Cerium-3+ can mimic the actinium-3+ with appropriate chelating agent, and then inappropriate chelating agent, the Cerium-4+ can mimic the thorium-4+, and means thorium-227. With very recent application, they also showed that the Cerium-134 allows for longer-term tumor targeting with DOTA-based antibody drug conjugates. And I don't have enough time to cover more details, but there's the 2 publications mentioned on the bottom of the slide. Since we're talking cerium, I don't want to take much time, but I want to also mention Cerium-139, which is a new product in the NIDC catalog. This one is much longer lived than 134, but the thought is, and I hope we can have a discussion on this, that there might be a possibility to use the Cerium-139, maybe for some proof of concept early on studies, if the customer is not ready for very short-lived Cerium-134 product. We produce it exactly the same way as the Cerium-134, only we come in at much lower energy for production. It's PN reaction, and you can see the final product, what it's look like, the spectrum. There is one really beautiful, very lower energy gamma emission peak. So it's easy to track the isotope, and the radionuclidic purity is excellent. And also the specific activity is somewhat lower than 134, which might not be viewed very positively as a possible surrogate, but I want to mention that the gamma line branching ratio is much higher, so there is a possibility to use much smaller amounts of this material. So I wanted to bring it up and hopefully see what others have to say about this possibility. To conclude my presentation, we produced evaluation batches and first production batch here at LANL, and we transferred our production and processing procedures to our colleagues at Brookhaven National Laboratory. And they were successfully produced their evaluation batches, and now we can provide year-round supply, steady supply of Cerium-134, and we produce it on Curie level with high specific activity and a radioisotope purity. Also, our collaborators demonstrated that the cerium allows for the long-term tumor targeting, and just as an outlook for us here at LANL, if there is a growing need for this isotope and we need to produce more, there is possibility for us to encapsulate it in more robust welded target shells, which can provide, which we can use higher currents for production and provide more Cerium-134, and also possibility for improved 134-139 ratio. And we can also suggesting that possibly if customers would like to do some proof of concept experiments, there might be a possibility to use Cerium-139 as a long-lived Cerium-134 surrogate. Just to mention briefly, Cerium-139 is typically used as mixed calibration standard, so it's also a very useful isotope for that. And this is a huge effort. There's a lot of people involved. Here at LANL, we have a large team of people to make this happen. I would like to also thank LANL colleague at Sigma for fabricating the lanthanum pucks, and colleagues at University of Wisconsin and Lawrence Berkeley National Lab for the first in vivo studies, and also collaboration on the cross-section measurements. So this is all from me, and I will stop sharing, and I would like to introduce Dr. Robert Flavel. He's from University of California, San Francisco, and the floor is yours.
Okay, thank you very much for the kind introduction and for the excellent background on the development of Cerium-134. And I'll just say on behalf of all the attendees of the webinar that we appreciate your efforts here on this endeavor. So the title of my lecture today is Development of Cerium-134 Lanthanum Pair as a PET imaging surrogate for Actinium-225 therapeutics in prostate cancer. Before I go any further, I'd like to highlight that the work I'm presenting today is primarily conducted by one member in my laboratory, Dr. K. Naidu Baba, who's an assistant professional researcher and has done really just a great job advancing this project. So Actinium-225 radionuclide therapy has demonstrated great promise for treatment. And this is particularly true in prostate cancer. For example, Actinium-225 PSMA-617 has demonstrated efficacy in the treatment of prostate cancer patients in small clinical trials. A lot of excitement in bringing this sort of treatment on into larger clinical trials. I think many of you will have seen this image before in the bottom left, but this is an example of a patient who had extensive metastatic prostate cancer, who was treated with lutetium PSMA-617, which is a clinical drug known as Pluvicto. This patient unfortunately had progression of disease after their first treatment. They were then switched over to Actinium-225 treatment and really had a marked response. You can see all this dark stuff here in the middle of the patient's abdomen subsequently resolved. So indicating essentially a complete response to treatment following actinium after progression with lutetium. And this has been replicated in some other patients and in small clinical series. So a lot of excitement around the use of actinium-targeted PSMA agents. Also in my laboratory, we're working on another target in prostate cancer called CD46. This is expressed across a variety of different cancers, including prostate cancer. And in some preclinical studies here, we saw some nice efficacy using a full-length IgG targeted against CD46. In this experiment here, we saw a really nice response following 0.25 or 0.5 microcuries of the actinium antibody with marked suppression of tumor growth and improvement in overall survival against the control groups. So I'm going to return back to these in the context of cerium imaging later on in the talk. Now, one challenge with actinium in particular is that it's difficult to directly measure the in vivo pharmacokinetics of actinium-225 labeled molecules. Why is that? Well, it's two reasons. So firstly, the daughter isotopes that produce gammas are francium-221 and bismuth-213. And in fact, they produce a relatively low fraction of these imageable photons, which could be picked up using SPECT imaging. The second factor is that the administered activities with actinium-225 is much lower compared to other therapeutic agents, such as lutetium-177, for example. So for this reason, actinium-225 imaging is not very practical, and this leads to challenges in implementing this isotope in the clinic. So a number of different approaches are being looked into this right now, and one of these is the use of radiolanthanides as companion surrogates. And in particular, the lanthanides, cerium, lanthanum, neodymium, and praseodymium have demonstrated promise in the regard. And of course, the title and objective of today's webinar is on the use of cerium-134 and lanthanum-134. I'll just point out that that it's not the only show in town, and a variety of other isotopes have been proposed for this purpose. But because of dissimilar chemistry and differences in isotopes that are common go-to PET isotopes, such as copper-64, gallium-68, and zirconium-89, don't work out that well for this particular purpose. So a lot of efforts on the use of different lanthanides. And interestingly, one thing that's particularly nice about cerium-134 is the relatively long half-life of 77 hours. And so therefore, it's been hypothesized, and the work was already nicely summarized by Dr. Mako, that the cerium-134 lanthanum pair may serve as a better PET imaging isotope for actinium-225 therapy. So cerium-134 does act as an in vivo generator. The parent isotope has a half-life of about 3.16 days and develops the daughter isotope, lanthanum-134, which is the positron emitter. Therefore, this exists in a secular equilibrium. So, if you had a hypothetical pure sample of cerium-134, you'll see the ingrowth of lanthanum-134. And we're going to return to this again later on in the talk. So in its initial incarnation, as developed by Dr. Mako and Rebecca Abergel and many other collaborators, there were two different chelators that were employed for this, the DTPA and DOTA chelators, which are very commonly used in nuclear medicine applications. These are very versatile chelators that can pick up a bunch of different isotopes. And in these initial studies, they looked at relatively higher molar ratios and higher temperatures for the radiolabeling kinetics. So our objective was to develop a robust labeling method for the chelation with cerium-134, which could act as a surrogate for actinium-225-based theranostic agents, and to use these developed methods for prostate cancer imaging using the small molecule PSMA-617 and prostate cancer targeting antibody YS5. So we looked at, in addition to the commonly used DOTA chelator, we looked at the more recently developed Macropa chelator for this purpose, initially developed by Nikki Teal and Justin Wilson. We rationalized, since the chemistry of the actinium-225 is cerium-134, and the Macropa chelator demonstrated efficient radiolabeling kinetics for actinium-225, that it could work well for this purpose for cerium-134 as well. So Dr. Baba initially started off with some model chelator studies using just DOTA and the Macropa free amine version, and did mock radiolabeling, or I should say model radiolabeling studies at 25 degrees pH 8.0, and monitored the reaction kinetics after 1 hour. And what he found was that there was relatively efficient radiolabeling with the cerium-134 and Macropa, even at low ligand to metal ratios, in fact, at 1:1 ligand to metal ratio, whereas the cerium-134 DOTA combination did require a little bit of higher ligand present in order to drive the reaction to completion. We tested the stability of these complexes in vivo. And so in this experiment, wild-type mice were administered either free cerium-134 metal, cerium-134 chelated to the Macropa, as well as the DOTA. And what we saw, and we compared it against actinium-225 itself. And then comparing the free metals, cerium-134 and actinium-225, we actually did see some significant differences here, particularly in liver, heart, kidneys, and bone. So that's interesting to note. But then when we took the chelated versions of these, we're showing the Macropa version, comparing cerium and actinium, essentially similar biodistribution. Looking at the DOTA conjugates, again, quite similar biodistribution, with one exception, that we did see a bit more bone uptake with the actinium version of it compared to the cerium. So overall, this demonstrated high in vivo stability of both the cerium-134 DOTA and Macropa, and the ex vivo biodistribution of the cerium and the actinium conjugates were largely similar. So with this, we turn to some applications. One that we have used is cerium PSMA-617. So this is, when labeled with lutetium-177, this is the commercial drug Pluvicto, which is used routinely for treatment of prostate cancer. So we use similar radio labeling methods here at 60 degrees in order to get the reaction to go to completion more efficiently to label the cerium PSMA 617. We found, again, in order to get a really great radiochemical yield, that the use of higher concentrations of the PSMA 617 was needed to get it to completion, and was able to get us a good result. We did some in vitro studies. We used the PC3 PIP model, which overexpresses PSMA, and compared it against the PC3 flu, which is a non-PSMA expressing cell line. As expected, the cerium PSMA 617 had good binding to the PC3 PIP cell line, but not in controls. And then over time, we saw gradual accumulation and internalization of the Cerium-134 PSMA 617. The KD of 32 nanomolar was in kind of similar ballpark compared to the lutetium and the actinium versions of it. We compared the cell binding to the actinium-225 version here, in this case, actinium-225 PSMA 617. That's the same thing that was administered to those patients earlier on that I showed you on in the first slide of the talk. And as expected, we say good cell binding and internalization in the case of the actinium PSMA 617 in the PSMA positive cell line. So with this, we turned to PET imaging. Here we used a dual xenograph model where we have one shoulder which has implanted the PSMA positive PIP line, and the contralateral shoulder implanted with the PSMA negative PC3 flu line. And then, as expected, we see accumulation of the isotope in the PSMA positive PC3 PIP, but not in the PC3 flu. And we confirm this in biodistribution studies. As expected, we see high uptake in the tumor in the PC3 PIP cell line, although I'll point out it does wash out over time. And really not a whole lot of uptake in other tissues, with the exception of the kidney. So we also looked at the full-length antibody, the CD46 targeting antibody. In this case, we labeled this with Macropa because we felt that these more efficient radiolabeling kinetics could be more useful for more sensitive applications like full-length IgGs. So NIDU applied the radiolabeling methods that he had optimized earlier with room temperature labeling for about 1 hour, and found good radiochemical yield of about 95% with good specific activity. One thing that's interesting to note about this, and it takes a little bit of getting used to if you haven't worked with this sort of isotope before, is this phenomenon that we saw, which is our crude reaction mixture after analysis on TLC looked something like this, where we see 95% purity or so. After purification on the PD10 column, we saw an apparent paradox, which is that the purity appeared to get worse. Now, in order to address this, we simply took this same plate and re-imaged the exact same TLC plate, I should say, 1 hour afterwards. And interestingly, this peak at the solvent front then disappeared. So what this peak is the free lanthanum. And because of the recoil effect, this actually does get ejected from the chelator. And if you're not expecting it, it can throw you for a loop. So just for people experimenting with this, if you see confusing things on your TLC or HPLC, you can wait a little bit and rerun it. So we did some biological validations of this Cerium-134 Macropa YS5. The immunoreactivity of it was preserved as expected, and we had good binding to the CD46 positive 22RV1 cell line, the KD of 3.7 nanomolar. And so therefore, this radiopharmaceutical could be synthesized effectively with 1-to-1 ligand to metal ratios and little or no loss of CD46 binding activity. We then went and did PET imaging studies in the 22RV1 xenograph model. We did various time points ranging from 4 hours to 7 days. And as expected, we see gradual accumulation in the tumor over the time with relatively low uptake in other tissues, with the exception of the liver. We then went and compared the biodistribution of the Cerium-134 and actinium-225 Macropa PEG YS5 versions. And what we found is generally similar biodistribution across most tissues, and particularly in the tumor, with two notable exceptions, however, in both the liver and the spleen. So indicating that there certainly is some complexity here that we don't yet have a great answer for. But that being said, This does enable imaging of this Macropa conjugate with similar tumor uptake. So looking quite promising for this PET imaging purpose. So I'll summarize the talk. So the radiolabeling methodology for cerium-134 is suitable for small molecules using DOTA. We found that the biomolecules benefit from more efficient Macropa chelate. The in vivo PET imaging reveals high in vivo stability of cerium-134 DOTA and Macropa NH2. The cerium-134 PSMA-617 has similar in vitro cell binding to actinium PSMA-617, and the PET imaging of cerium-134 Macropa PEG4YS5 showed a high tumor uptake at 7 days post-injection, and similar biological findings for the PSMA derivative, good uptake at early time points with washout at delayed time points. And the ex vivo biodistribution was consistent for the cerium-134 and the actinium-225 Macropa PEG4YS5 in tumor in most tissues, with the notable exception of the liver and the spleen. So therefore, the cerium-134 actinium-225 pair could serve as a theranostic pair in prostate cancer. I'd like to conclude by acknowledging all of the collaborators and lab members, again, particularly K. Naidu Baba, who led the effort. I'd like to also additionally especially acknowledge Dr. Henry Van Broeklen, and we are co-PIs together there on this project. I'd like to thank the DOE for funding this work, and then, of course, for production of not only the cerium-134 isotope, but also the actinium-225. So I think we're going to be holding questions for the end. If there's anything that's not addressed during the discussion section, please feel free to shoot me an email, and I would be happy to discuss further. And thank you very much for your attention.
Thanks so much, Robert, for that. Was really interesting. I love to see all the data with cerium-134. And I would like to welcome Timothy Haysted from Duke University to present the next talk.
Hello. Can you hear me?
Can you put it into the presentation? Yes.
Yeah. Oh,
perfect. Thank you.
Let me go back to the start. Yeah. Well, thank you. I very much appreciate being able to talk at this meeting. You'll see we're coming from a different angle than anybody else, I suspect, talking today. And I've already learned something about different chelates available out there. As you'll see in a minute, this has become an issue for us. Let me see if I can get rid of my bar. I don't know if I assume you can't see that. I've worked on this protein that's called HSP90 for over 30 years. Since I was an assistant professor, we were the ones that discovered it was an ATP binding protein back in the year, before that was an unusual finding. And what we're interested in is developing it now as a PET agent, as a diagnostic target. And what I'd like to do today is tell you how we're doing that, and hopefully you guys can help us. It's really blossomed, this program, and we now are not only involved in some cancer work, but we're starting to blossom out into infectious disease, which might become apparent in a moment. Let me get this thing to move. We actually my background is somewhat in biochemistry and chemical biology, and we developed a screen. We actually produced the first fully synthetic HSP90 inhibitor in the early 2000s, and made a drug that ends up going into clinical trials, which was acquired by Pfizer. We did it in this garage in downtown Durham. When the company was acquired, we went and got interested in another form of HSP90 that we discovered using our HSP90 inhibitor. There's the pills right there, which associates expression of the protein with malignancy on the surface of cells. It started with a close colleague of mine, Len Neckers at the NCI, who told me antibodies to HSP90 could block cells migrating in a scratch assay, and he thought it was involved in metastatic progression. So we got interested in that. And because we are sort of a chemical biology laboratory, we decided to make a ligand that target based on our original co-structure of our HSP90 inhibitor, which we tether to an imaging agent. And we start this whole program at the cell biological level. So the idea is that we made this probe thinking that we would attach things like fluorescein that would not enter cells by diffusion easily. And whereas a drug, a small molecule like our original HSP90 inhibitor, can diffuse through the membrane. So we made these things, and we did it in a classical biochemical way. You have your inhibitor, you figure out where you attach the ligand so you don't interfere with binding to the target. You make an affinity column. This is recent work we can show with new versions that target a bacterial form. It rather cleanly pulls out just the target. That's true of every cell type you've ever looked at. And then you can go and merrily add things around the outside. And the next job is to see if they go into cells, obviously. We worked on these a lot. These are great tool compounds to study this protein, and we've given these out over the years to various labs. But we've been trying to argue with our colleagues in the field that we think HSP90 is a great diagnostic target and target for therapy using PET type approaches. The first piece of evidence that we had that our probes behaved in an interesting way was that we added the fluorescein version, and you can see in this study these are different cancer cell lines, and you can clearly see, consistent with Len Necker's original observation, that only the most these are 4 sets of breast lines and 1 non-breast line that won't form tumors in mice. These do, where we can see a massive uptake of this probe into these cells. We go do fancy things like high 3-dimensional confocal microscopy to look at what's the mechanism of entry, and we can see that the drug, when it is applied to the surface at even at low concentrations, will form these aggregates. And when you look deep into the cell, they look like they go in, and they form these little aggregates. And we think they dissolve with the material inside. And then the internal pool, what's interesting about HSP90 is the internal pool of HSP90 is about 1 to 2% of the cell protein. And this causes long retention and accumulation. So when we measure the concentration of the drugs in these cells, it gets up to micromolar. And we published this, and what we see is, hypothesis is that normal cells don't express the surface form of HSP90, which we refer to as ectopic or eHSP90, because we think it's on the surface. And we think that when you bind the drugs with the tethers on it, there's some form of aggregates form, and they get internalized. And all kinds of genetic studies were done, siRNA silencing to disrupt the process. And I think 1/3 of the libraries, the siRNA libraries involved in transportation, would block this process, whatever that means. But it seems to be like an active process. We then go on to ask the question, how specific is this? So here's a breast model tumor line where we inject the floor probe up. And in this particular example, we then slice the animal up 700 times and then imaged all the slices. And there's a nice movie that comes with this, but it takes a while to load. Essentially, the take-home message was it seemed like only the tumor was taking in these probes, and you could see how the probe was being eliminated via the bowel. We then proceeded to move this down a clinical path and towards an IND. I'm glossing over a lot of things, obviously, but we made some inactive analogs and looked at the pharmacokinetics in mice with our colleagues at the Lyerly lab and with Takuya Asada, who did this work, in which we injected the active form. This is called HS196. It's a deep red probe, and compared it to HS199, which is an inactive version. And you can, it's a true analog. You see this long retention in the tumors of the mice with the active form, and it's cleared quite quickly in the thing. This is using an IVIS machine. These are only flank tumors, obviously. This did progress to a clinical trial in men with who were slated for prostatectomy. This group of individuals were known to have, they were PMSA negative, and we had, with a variety of cell-based studies, decided that this probe would recognize those cells that form prostate cancer. It's different from the PMSA that Dr. Flavell talked about, which is about 80% of men who have prostate cancer. There's a small minority, fortunately, that don't respond to PMSA positive treatments, such as the wonderful 117 version that's in clinical practice that Dr. Flavell mentioned. But you could see histologically uptake into these tumors. So what happened was this compound was given 24 hours before surgery. Then the tumors were resected, and then the pathologist made his assessment, and then agreed that these, and then the same slides were looked at by fluorescence, and agreed that these areas of uptake were, in his opinion, regions of aggressive disease. We did single cell seq on those, and if you see the paper, you'll see that the main phenotype of where the probe is taken up is in cells with a neuroendocrine phenotype consistent with PMSA negative disease. So now where do you go with this? It's obvious we'd like to make a PET agent, and we started with iodine. And I should mention we've just got our own PET machine. It's just being delivered. So we're early in the game here, and I'd collaborated with UNC on this, but we did make an iodinated version, and then looked at it in an animal model. This was an animal model that is a spontaneous tumor model, which you have to do about 10 mice before you get one that does form a secondary mass. But in these studies, we compare the F18 FDG PET image the day before. You can see the usual distribution of the glucose molecule, and then we did pick up a secondary mass. What was extraordinary was there was no uptake anywhere else. The only disadvantage of this probe is it goes out into the bowels, and that's the gallbladder right there. We did verify this histologically as a secondary mass. There was uptake in the primary tumor, but it didn't show up as well as the secondary. But iodine is not something we want to work with. The production of the molecule iodine involves stanylation, which involves tin, and it's from a radiopharmaceutical point of view, it's not the most ideal, and even for chemists to make, it's not ideal. So we really want to switch to chelates, and we're naive in this business, obviously, but we got DOTA, added it to our ligand, and then fortunately got we've only had 2 batches of your Cerium-134, but we found it was very effective at keeping our probe out of the cell. The free metal will go in quite easily, obviously, and you wash it and you caught some sort of equilibrium, but this was rather good at keeping it out. This just compares in the same day just to make sure those cells were good with a floor version, that the cells had a surface form and the inactive molecule didn't bind. Whoopsie, what's this? Jump over here. I'm trying to get out of this. So one of our major efforts, and what we'd really appreciate, is some help here. And I wrote down some chelates that I hadn't heard of. I had heard of HBED-CC, which is the one that's attached to the PSMA molecule. My wife is a radiologist, so I hear about this. And we are currently trying to synthesize that to get full synthetic control over it. I had not heard of some of these other ones, so I'm interested in looking at those. Just a quick touch on what we're doing next, and that is to do with, we've also been funded heavily to look at infectious diseases because our same target is actually conserved in a number of bacterial organisms. In fact, all bacterial pathogens, parasites, yeast express the protein, and we've been asked to look at that, which we are starting to do, and we can get our 4 probes in, but we'd like to get our tethered versions. The reason we're able to do this is through the power of chemistry, obviously, is to we can tailor the ligand, so we can target it to be species specific. To cut a long story short, but it's very exciting. I actually don't know why I'm showing you this. I'll skip that. But we've seen the same issue with the DOTA version of when we link it to our Borrelia specific, which is a Lyme disease model we use. Again, it's not traversing, whereas in the same experiment you could get quite nicely by fluorescence this compound. It carries vertephorin, which can actually kill the organism with. And that's another program. But what's interesting about this is we're also interested, and we work with one of the physicists who's interested in capturing Cherenkov radiation and transferring that energy to our vertephorin version to generate reactive oxygen. Interestingly, when we tried to, put the C34 in with a ligand that we designed to actually capture that based on altering some double bonds in this structure, could enhance the Cherenkov effect, at least in a scintillation counter, and actually kill the organism in culture. This form of Cherenkov radiation generated by positrons, my understanding is it's a weak form, and X-rays could generate maybe tenfold the signal, if not more. I think it's nanojoules of energy, Cherenkov versus micro, I believe. So this is obviously a massive effort on a lot of people in my lab and collaborators around the world, and that I'm very grateful to. And my funding is listed here. Thank you. I shall stop sharing.
Thank you so much for interesting presentation. I think there's definitely from other presenters information about other ligands and several publications by our collaborators too. And the last presentation today will be by James Kelly from Weill Cornell Medicine. Oh, please take the floor. Thanks.
Okay, thank you very much, not only for the opportunity to present, but also to my fellow panelists for outlining the background of Cerium-134 so concisely, so that I don't need to revisit any of that. And you'll see there's a lot of thematic overlap to our objectives, as I think is probably to be expected, given the wide interest in developing actinium-225 radiotherapeutics and trying to identify possible imaging and dosimetry surrogates. So again, just to quickly give an indication of where we're going here. So our objectives were to compare the labeling, purification, and QC methods for Cerium-134 and actinium-225, and in our cases in the context of small molecule PSMA ligands. And then we were also interested in comparing the in vitro and in vivo stabilities of the 2 compounds, and finally the tissue distribution. And before I get any further, I want to highlight my very talented postdoc, Dr. Wacker, who not only performed all of this work, but was heavily involved in conceiving of these experiments and really has been driving our preliminary program forward in this area. So we did some initial radiolabeling studies here where we were comparing the labeling of a macropa PSMA ligand, which in our nomenclature is RPS-088, and a DOTA ligand, which is RPS-063. These are otherwise structurally identical. And just looking at conditions for labeling with Cerium-134. So as is shown in this table, we looked at both acidic and basic mobile phases. In the case of the macropa containing compound, we looked at room temperature. And in the case of the DOTA compound at 95 degrees C, And as the graphs on the right will indicate, in both cases, we get very high levels of complexation after just 5 minutes, showing that labeling with cerium-134 is highly practical for small molecule ligands. For QC, we used a combination of methods. Primarily, we were looking at radio TLC, and we looked at 2 different mobile phases. So using this 50 millimolar citrate mobile phase, we found that our labeled complex stayed more or less on the application point, while the free or unbound radiometal traveled with the solvent front. When we switched to a 1-to-1 ammonium acetate DMF mobile phase, the reverse was true. So we had migration of our labeled complex with the solvent front and retention of the unbound metal at the application point. And we thought it was important to look at both of these because we wanted to eliminate the possibility of some colloidal species or some other unidentified cerium compound that might stay at the application point and might be overlapping in signal with our labeled compound in the 50 millimolar citrate. So this way we were able to assure ourselves that we in fact had a clean compound. And these 2 mobile phases give exactly the same results with actinium. So in this case, we're imaging the TLC plate by phosphor imager rather than by a TLC scanner, simply because we didn't have enough activity for the TLC scanner to detect the signals. But again, in the case of the ammonium acetate DMF method, we see migration of our compound. And in the case of the citrate, we see it staying at the application point. And as has already been pointed out, these analyses must be performed at least some time after approaching secular equilibrium. So in the case of cerium-134, we were doing our analyses 1 hour after developing the TLC plate. And in the case of actinium, it's 2 hours after developing the TLC plate. It's also possible to do the QC from the HPLC. And in this case, Anya has developed a really ingenious method of collecting fractions from the HPLC, spotting them on a grid, and then viewing that grid on the phosphorimager and using those data to reconstruct the HPLC signal. So what you see on the top here, where my laser pointer is the radio TLC trace from the HPLC. And what we see beneath it is the what we're calling the true chromatogram. So the chromatogram produced after secular equilibrium was reached. And just as Professor Flavel pointed earlier, you see disappearance of the peak at the front, which is the lanthanum that's decayed out. And we see this is true both for the macropod containing compound and also the DOE containing compound. So on to our in vitro experience. So here we are looking at the relative stabilities of these compounds, both to serum of human and murine serum, and also a challenge with DTPA. So for our serum stability studies, we incubated at 37 degrees C in either human or mouse serum. We assayed the aliquots at various time points. And in this case, I'm showing just the results from the 50 millimolar citrate TLC, although we also did the experiment in the mixed mobile phase as well. But for the sake of time, I'm just showing by a single analysis method. So depending on whether we use the phosphor imager for quantification or the TLC signal, we got slightly different results, but they both agreed that there's some decrease in serum stability, or there's some loss of cerium from the complex in serum over the course of 72 to I'll say 168 hours. And depending on which method you use, that loss is anywhere from about 85. So it's anywhere from about 85% stable to perhaps a little lower than that. And by comparison, we think the DOTA complex is slightly more stable. So in this case, again, these are preliminary studies and we've only had the opportunity to do phosphorimaging based quantification. But if we compare the top and the bottom, so these are analyzing via the same method, we see it's comparable, but maybe slightly higher stability of the DOTA containing compound. And this is true in both human and murine serum. When it comes to actinium, so actinium, we observe the inverse. In this case, it's a more clear difference in that the macropod compound is highly stable in serum, whereas the DOTA compound is much less so. In the DTPA challenge experiment, we more or less reproduced the results of the serum stability, in that with our macropod containing compound, it's mostly stable until we get to about 1,000-fold molar excess of DTPA, at which point we do start to see a significant level of decomplexation. And in the case of the DOTA compound, there's a little bit of noise in here that obviously we hope to clean up in future experiments, but the take-home message is that the final amount of complex material is comparable to the initial amount, and that's true irrespective of how many molar, fold excess of DTPA that we have, all the way up to 1,000. And once again, when we compare it to actinium, we see again the inverse. So in the case of the macropod containing ligand, we see no decomplexation. And in the case of the DOTA containing ligand, even at relatively low levels of DTPA, so even at 1 molar concentration or 100 molar excess, we already begin to observe a little bit of decomplexation over time. Although Anya has asked me to point out here that in this particular experiment, we were starting with a relatively impure compound. And so it's possible that there may be some artifacts that are interfering with our measurements here. So of course, we'll need to repeat this with a more highly pure starting material. Finally, to move on to our in vivo experiments, and these were experiments conducted in an LNCap tumor model. So this is a prostate cancer model, and we administered the mice with about 4.5 megabecquerels of the cerium-containing ligand. In this case, we at present only have imaging data for the macropod-containing compound, so that's the RPS-088. But we imaged these mice at 2 to 72 hours post-injection with a 30-minute acquisition. We did image-based quantification by using a 1% injected dose standard to help us get the values. And based on the many previous studies we've done in our prostate cancer models, the biodistribution is as expected, with the primary sites of uptake being the tumor and then the excretion through the kidneys and bladder. However, I do want to highlight here that you can see liver, particularly at the 2-hour time point, but also a little bit at the 72-hour time point. And this is in contrast to what we've seen with previous ligands, which don't clear by the hepatobiliary route, and was an indication perhaps of some in vivo decomplexation of cerium, as it was shown in the previous talks that free cerium accumulates in the liver. So finally, we sought to do initially an inter-subject comparison between our cerium-labeled compounds and the actinium-labeled compounds. We injected the mice with either 3.5 megabecquerels of cerium-134 RPS-088 or 50 kilobecquerels of actinium RPS-088. And at this point, we've only done a single time point biodistribution at 24 hours post-injection. We had 3 to 5 mice per group at this time point. And once again, the two track very nicely. The tumor here, there's just there's no statistically significant difference, but rather this just reflects the biological variability of the LNCap tumor model, which tends to grow out rather heterogeneously in the animals. Where there is a really, and this both statistically and perhaps biologically significant differences in the kidneys. And we see much lower kidney uptake of the cerium compound relative to the actinium. And our working hypothesis, which we still need to test, is that there's the clearance, the decomplexation of cerium leads to its accumulation in the liver at earlier time points, but that this has cleared by 24 hours. So we no longer see any difference in the liver, but and the deficit in the kidneys simply reflects the level of the extent of decomplexation that's happened to now. But as I say, we'll need to test this by repeating the BioD at earlier time points. And finally, we were working on an inter-subject comparison. So this would be one way of trying to remove biological variability from the equation by actually co-administering the cerium and actinium compounds to the same animal. And so we performed the same experiment. In this case, we only had 2 animals so far. And again, we sacrificed some 24 hours post-injection. And we counted the tissues beginning 24 hours after harvest and continuing up to 328 hours. And what we can see, and again, we were scaling the activity based on known standards that we prepared. And obviously, it's reassuringly comparable between 24 hours and 328 hours, and between mice as well. So the kidneys are slightly different, but not significantly so, and all the other tissues look identical between the two mice. What we're really interested in doing is seeing whether it's actually possible to discriminate between or distinguish between perhaps the contribution of cerium and the contribution of actinium to this particular tissue activity. And so we've been using a graphical method to estimate what this might be. We had prepared standards, mixed standards of actinium and cerium ranging from 0 to 100% cerium, and sort of back calculated using decay equations what the starting activity was at these standards based on observed measurements at different time points. And as the graph in the bottom right-hand corner shows here, the measured values, which are here in the maroon curve, match quite closely to what the calculated activities should be. There's about a 3 to 5% error from this method. And so when we applied this to the biodistribution data, we found that it actually recapitulated our inter-subject results very nicely. So if you recall from a couple of slides back, when in fact the kidneys looked even higher, or the kidneys look fairly comparable, but now when we actually separate out the activity, we can see that the contribution of actinium is much higher than cerium. Again, this is consistent with what we saw before. There is a higher background level of actinium in general. So if you look at the large intestine, the liver, even perhaps bone, and that may be due to the sort of 3 to 5% error in the cerium. So we're still trying to optimize this method, but certainly our preliminary experience suggests that it will be possible using standard graphical methods to actually determine how much of the activity is due to one isotope or the other. And again, this is just a reminder of what the inter-subject data looked like, and you can see that the intra-subject looks very comparable. So in summary, we found that both macropa and DOTA rapidly complex cerium, either at room temperature or 95 degrees, that the DOTA complex is stable and in cerium and to DPA challenge. The macropa decomplexes a little more in those conditions, and that's inverse to the complex stability of actinium-225. So it just has to be taken into account when doing the in vivo experiments. Sure enough, perhaps because of these differences in stability, although, as I say, we haven't yet done the imaging with the DOTA-containing ligand, but we do see some additional signal in the liver from our cerium macropa compound that hints at in vivo instability, and that this decomplexation may lead to different doses absorbed by the excretory organs in comparison to actinium. So that's something that might have to be factored into dosimetry calculations moving forward. But I think one of the things we're excited about is that it will likely be possible to disentangle the contributions of cerium-134 and actinium-225 when co-administered to the same animal. And just to quickly acknowledge, the people who've worked on this. So from my group, Drs. Faker and Ascona and Lee, Dr. Amor Coraza from Ratio Therapeutics, who's helped not only in supplying the cerium for us, but also in designing many of these experiments. And of course, to all of the people at the NIDC who've been able to provide us with the cerium-134. We've been thrilled to be part of these initial discovery efforts, and we'd be very happy to answer any questions that anyone from the audience may have. And with that, I'll stop sharing.
Thanks so much for interesting presentation. I guess right now this is a conclusion of all the presentations. So we would like to invite questions for any speakers. And I'm trying to, I got my video going. I would like to ask all the speakers to turn on their videos as well. Thanks so much. And it's look like I'm gonna check the Q&A. Oh, here. There is a first question. Is there a way to produce Cerium-134 free of 139? And the short answer is that we can maximize a little bit the ratio of 134 versus 139, but unfortunately the production cross-section for 139, you can look in one of the papers cited that it decreases, but it doesn't go completely to zero. So unfortunately, there will be always a little bit, but you know, there might be a little bit possibility to tweak to improve the ratio. Yeah, I have one question for Dr. Flavel. Like, I was wondering when you saw if you have any thoughts on there is similar uptake, but it's look like that there is a little bit difference in liver and spleen. I'm wondering if you have an idea what might be going on in there or some hypothesis on that.
Yeah, I think the simplest explanation for this is one that Dr. Kelly alluded to, which is that probably at later is that there is a slight instability and that there is probably some decomplexation of the cerium at later time points. We weren't able to detect that at the early time points with just the chelator, but if, you know, for a full length IgG that's hanging out for days in serum, it's probably slowly releasing it. The second thing I'll add that's interesting is since the cerium itself is acting as an in vivo generator, it's producing the lanthanum, and because of the recoil effect, the lanthanum is being ejected from the chelator. So for, you know, agents that are in the blood pool, you'll see the apparent reaccumulation of the lanthanum in vivo in the liver as well at the early time point. So a lot of complexity here going on compared to your typical, you know, your typical PET imaging agent where it just, you know, goes to its target. So yeah, I think both of these things are very interesting and merit further study. Yeah.
Thanks. Okay, there is a question again on production, so it's more for us. What about alternative targets? I would say that because of you know where it lies, like if you look at the isotope table, it's kind of hard to get to Cerium-134. So, you know, I think early on, I'm sure our colleagues look at different possibilities, but I think the proton route is really the only one which provides high yields for Cerium-134. And I don't know, I might have a colleague Ellen on the chat which might add a little bit more background on that if possible, but you know, I would say the short answer is really no, that this is the best way to produce the Cerium-134. And there is a question. Nice work. I have a question related to the lanthanum progeny. Is the isotope utilized for the PET imaging signal? Is there any corrections to implement for the cerium lanthanum possible PK differences in vivo? I'm not sure I'm totally understanding this question.
I can step in if you'd like.
Okay, yes, please.
So I think this is similar to what Dr. Flavel was just talking about, which is that you know, it's possible that the at least some fraction of the lanthanum will not be bound to the molecule anymore at the time of emitting the positron. And so the question is, how much redistribution do we expect, I think? And of course, that's a problem with actinium as well, right? So actinium with its various daughters, you know, and those daughters can also redistribute. So that was when I was presenting at the end of our talk about kind of trying to disentangle the contributions of the different isotopes. that's sort of what our long-term aim is to address, is to see if we can take a tissue sample and actually measure the degree of redistribution of the cerium-lanthanum pair compared to the actinium and its daughters, and see, you know, in a given tissue, how far do those actinium daughters go? How far do the how far does the cerium-lanthanum pair go? And see, you know, really how well are they sort of mirroring each other. But I think there's almost certainly going to be a need to do some form of correction, but I think it's too premature to say exactly what that will be. But I think that's one of the questions we're trying to answer.
Thank you. I was wondering, so if you're thinking about, you know, the possibility the lanthanum, right, getting ejected and maybe redistributed, I wonder for some initial studies, would any of you consider the cerium-139 and possibility of SPECT imaging for that one to see, because that's a longer lived isotope, to kind of help you to distinguish the biodistribution cerium versus lanthanum for the cerium-134? Any thoughts on that?
I would say half-life's a bit too long. If I worked on that here, I think my rad safety people would be angry with me. I have to keep things for 10 half-lives.
Ultimately, it decays.
You know,
it's not many years, but yeah, I totally agree that it's a little bit longer life. I was thinking really for some proof of concept studies, if it would be, you know, at all useful to use this isotope.
I think the cerium is 2 days is a good length of time. It's you know, you're going to get a signal for 4 or 5 half-lives, I think, depending on what your stoichiometry is of labeling and bump it up, I guess. But no, that's I think for any radiopharmaceutic, you want that half-life to be. How that would translate to humans, I don't know. You know, whether that would be useful in humans, probably with long-term tumor, in conjunction with actinium-225, you know, to see you're getting tumor regression.
Any other comments on this from other presenters?
I think it's certainly an interesting idea, but yeah, I certainly agree that the long half-life presents a challenge. I haven't thought of it yet. I'm not sure exactly how much activity we'd have to. It looks the energy is appropriate. It's a good energy. It's sort of close to technetium-99m. But is the abundance of the photon high for the cerium-139?
Yeah, gosh, I think it's on the 80-ish, 90% range.
That's actually pretty close to technetium-99m,
Yeah.
which is a great isotope. Yeah, the half-life is a tough one for sure. From, you know, it does present a radiation safety challenge, but I think for, you know, that's a addressable challenge. Certainly for preclinical studies, it could be useful for a validation sort of experiment.
That's what I was going for.
Sorry, I was going to say you probably get pharmacological clearance too of your agent, right, which will clear it away. It's just as long as you keep all the urines, I guess.
No.
I don't know.
Yeah, definitely not for human use. We want to limit the long-lived isotopes for sure.
Yeah,
but you know, for some preclinical studies, I was thinking that perhaps, you know, it could be valuable for some longer-term studies, and, you know, removing the uncertainty on, you know, having the in vivo generator, the cerium-lanthanum pair, right? It's gonna be only cerium.
Yeah, no, it's very useful for pharmacokinetic stuff that you're trying to look at different analogs.
We got a question on is HSP90 a target for a specific cancer?
No, there's very broad. Almost every metastatic form expresses that we've looked at, from glioblastomas to breast and prostate, pancreatic. It's expressed on the surface. It's also, you know, it was used to treat solid tumors as a free drug entered over 50 trials in the US, and the first one was approved in Japan as a drug. So, but we think it's an excellent target for as a PET agent simply because of the biomass of HSP90 in a cell at 1 to 2%. You know, that could matter from a signal to noise ratio. You could really play with the pharmacokinetics widely. The amount of radiation perhaps you have to deliver won't be as much if you could really pack it in there. So that's our hope. That's why we're interested in the PET, and really committed now. With $1 million machine just showed up, so I don't think they'll fire me too soon. Hopefully not. I wouldn't blame them, but it would. But yes, there'd be pretty much every cancer you can look at. Oh, apart from liquid tumors, I should qualify that. Actually, certain B cell lymphomas don't seem to express the protein. That's right. Yeah.
Quick question for you guys who already used Cerium-134. How do you like the form you provided? And it's provided in a hydrochloric acid. Like, would there be interest for other matrix? Would there be interest to provide it as a solid similar to actinium-225? Any thoughts on that?
Can I just chime in there? Could you put it in a vial that's easy to open, that doesn't involve, they put this thing around the top, and it doesn't even
you have to use the crimped up vials for the liquids for safety and for shipping perspective. I'm not sure we can do anything about it right now, but we can certainly look.
Yeah, we have some toxins that we work with that have this crimped little tear off piece.
Yeah. You can use a decrimper to open the vial safely, but it's additional tool. But I believe the actinium-225, because it's provided as a solid, has a screw top lid. So there might be a possibility, but then I think it might need to be a different form, but we can verify. We have shippers on our team who help us with all this logistics.
Yeah, it's a trivial matter, but you get it on your fingers when you're trying. And if you want to go back and use the vial, you can't put the lid back on. I mean, it matters, unfortunately, because it's a contamination issue, and it sounds trivial, but it's actually a bit tricky.
Dr. Flavel, how do you retrieve the cerium? Do you have any issues with that from the vial? Any tips?
That is a good question. I know I have not heard about any issues with this. I'll say I'm not doing it personally. I'll defer to Naidu. He's typing stuff in the chat, so maybe he'll be able to comment on it. But I haven't. I will say I have heard a little bit about that there could be some. It might be more convenient to have it as a solid, as an alternative, and there has been maybe a little bit of issue in terms of like variability in the concentration, et cetera. So I think that is something that could potentially be useful for the community to have it shipped as a solid form, similar to the way we're receiving the actinium.
And we have a question in the chat. The liver uptake for Cerium-134 actinium-225 RPS complexes show similar in ex vivo bio-D. However, the actinium-225 actinium complex is more stable than Cerium-134 complex in vitro studies. Then the liver uptake may not related to stability of the complexes. Any other possibilities?
Yeah, I mean, certainly it's hard to make any sort of fully supported hypotheses based on our single time point. Certainly another possibility could be that the size of the complex influences it. I mean, we expect, since both are macropod, that the overall charge should be the same, so that shouldn't drive any difference. But there's always the possibility that it's something we haven't considered yet. I think we'll have a better explanation once we've looked at different time points to see whether we really do see differences in particularly liver and kidney over time.
Awesome. We have a reply from Naidu, and that was his question actually as well, that they do not have any issues with the supply of cerium in 0.1 molar hydrochloric acid. He's not mentioning if about the vials though.
Well, I do it. That's the thing, because I won't let my students touch this. You know, there's paranoia about radioisotopes. So I'm only speaking from experience. So it's just anyway.
And there is also a note from Naidu, but nowadays we're not getting cold mass content. But I'm wondering if Naidu, can you explain more? Like, I'm not sure what exactly do you mean by this? I'm wondering if you can type that quickly.
Maybe it's the ICP-MS results. Are you guys measuring them on every batch?
Yes, we do. And, you know, to point out, we did supply some batches, and because we have alternate run cycles between us at LANL and Brookhaven National Laboratory, we try to have the specs very similar, but our target design is slightly different because of the different machines. So I'm wondering if they have maybe a little bit lower cerium content. I've seen a few colleagues from BNL if they might be able to comment on real time, but we can definitely have a look at more recent batches. We did compare all our evaluation batches. We did 2 at LANL, they did 2 at Brookhaven, and they were fairly similar with the specs, but they produced several batches recently, so I didn't have a chance to see all the data. So I'm wondering if they do a little bit different lengths of irradiation, and if maybe, and I believe their target is slightly smaller. And of course, the stable cerium, you know, we acquire as pure lanthanum as possible, but and with cerium levels on ppm, which is the best what we can do, it's actually really hard to get very pure lanthanum material. We couldn't go with commercial suppliers. There was too much stable cerium. There was hundreds of ppm. So we worked with colleagues at Ames National Laboratory who are do magic on lanthanides purification. So to acquire very low cerium lanthanum, but then if you go with a little bit bigger target, then you have the more stable cerium in there. So he is replying, cold metal mass content of lanthanum and cerium for calculating the ligand to metal ratio. So earlier, cold metal mass content for cerium and lanthanum was provided in the C of A. Does it mean that it was not provided the cerium lanthanum content recently? I'm wondering. We do provide it on ours, and I believe we received material from our colleagues as well to just look at the specification, and they did have the stable amount, I believe, on the first C of A, but I haven't seen it recently, and that's definitely something we can look at and provide for easier labeling. We cannot, I think, assure it will be within similar ranges, but you know, when you run the separation, I've seen it myself when I was developing the process that for example, the lanthanum, we start with tens of grams of lanthanum, we get to micrograms, which is a really good decontamination for one separation step, but it varies slightly how much residual lanthanum there might be. So providing this on the CFA, I think is a good idea, and we'll work together to see if we can provide it in the future. Alrighty. Any other questions from the presenters for each other?
Sure, I can ask one for Dr. Kelly. Actually, I was curious about your experiments where you're, trying to compare the actinium and cerium in distribution in the tissues. What kind of detection method are you using there? Are you using like a gamma counter, or what's the other, high purity germanium source, or what kind of detector are you using?
Yeah, at the moment it's just gamma counting. So we're just repeatedly counting the samples over a period of time against the known standards and using that to plot the curves. But I think, longer term, the idea would be to use a sort of hybrid of either kind of imaging techniques or alpha camera type imaging supplemented with gamma counting and or like jelly detector type spectroscopy to really get a full picture. But like I said, we're really in the early phases and just trying to establish some element of feasibility. But I think hopefully that some of these methods will prove useful as we progress to other detection methods.
Yeah, I have some questions to do with sources of chelates. I'm sort of madly working my way through different types of chelates. We have DOE, of course, and we've tried to grease that up to get it to go into cells. And we've just completed the synthesis of HBED-CC just to have a source of that, because it does go into cells. But that papers I've seen on that have about 10% bioavailability, they claim, which is better than DOE, which others also repeat that they can't seem to get that to enter cells. But sources of, I can look it up online now. I've heard the names, but a DTPA and the macro PA. I'm interested in those, and where you guys get those from.
So DTPA is definitely commercially available.
Which one is that? DPTA.
Okay. That's one of our, very, very vanilla sort of commonly used chelator. So DTPA definitely commercially available.
That binds all the lanthanides fairly well.
I have not tried that, but certainly in that initial publication, it was used. Yeah.
Okay. What about macro?
Macro pi, to my knowledge, is not commercially available, and we had to synthesize it in the lab.
Oh, okay. And okay. Does that show good bioavailability even on its own, or just it has to be linked to something? Just the metal complex clears out very rapidly. It doesn't seem to have much binding in normal tissues. It just gets rapidly excreted through the renal route. Yeah, is it? Well, that's good. In cells and things, is it going in there in any way? Or,
we see a lot of internalization when we link it to antibodies or like a PSMA ligand, for example. Yeah.
Yeah. Okay. Thanks.
I just want to mention that all of this recording will be available, and all of us had manuscripts links on the bottom of the slide. So that's a great way to get through the video. You can pause it and you can get the link to all the manuscripts, which several of them has information on different ligands. I have a quick question regarding the imaging. This is kind of outside of my field, but you mentioned the alpha camera. How does it work, and can you differentiate between different alpha energies, or basically you're imaging all alphas? Because of course, actinium has several alphas in the decay chain.
So I confess it's a little outside my area of expertise as well. My understanding is that there's some degree of distinction between alpha energies possible, but different devices have different levels of sensitivity, let's say, to those. So my expectation, at least in terms of how we're kind of moving this forward, is that it's not going to distinguish between the parent alpha emitted by actinium and the subsequent daughters, but it will distinguish between the alphas and any other form of radiation that might be present.
Got it. Right. I think we are reaching at the end of the recording and the session. I don't see any more Q&A questions, so I want to thank again all the presenters again and for nice discussion. And I would encourage everybody who else has any interest in these isotopes to reach out to us or reach out to NIDC regarding the availability. And yeah, I'm looking forward to more research on the cerium and seeing it application in the clinic. So thanks so much, everybody. It was my pleasure to host this seminar.